Hey everyone, I'm Roshana Flauta-Mesa, and I'm here to talk about colorectal cancer, or CRC, and its relation to the gut microbiome. Colorectal cancer is a common cancer that affects the colon or rectum. It is the third most common cancer in the United States and is a leading cause of cancer-related deaths. Signs and symptoms of CRC can include changes in bowel habits, blood in the stool, abdominal discomfort, unexplained weight loss, and fatigue. Now let's talk diet and lifestyle. Here are 4 dietitian and lifestyle tips to assist in the prevention of CRC. Number 1. A healthy diet rich in fiber, whole grains, and low in red and processed meat can reduce the risk of CRC by promoting the growth of beneficial bacteria that produce short-chain fatty acids, or sometimes referred to as SCFAs. Number 2. Regular physical activity can also lower the risk of CRC by increasing the abundance of beneficial bacteria like Akkermansia muciniphila and SCFA-producing bacteria in the gut. Number 3. Maintaining a healthy weight through proper diet and exercise is crucial, as obesity has been linked to a decrease in gut microbiotic diversity and increased risk of CRC development. And finally, number 4. Avoiding risk factors such as smoking and heavy alcohol consumption can also help prevent CRC by maintaining a healthy gut microbiome balance and reducing inflammation in the colon. You're probably wondering, who is more likely to have it, how do you get it, and how is it common? But don't worry, I got your back. Let's start with the who is more likely to have it. Individuals with a history of smoking and colorectal cancer within their family history are most likely to develop this disease, as well as those with a serologic response to Helicobacter pylori. Now on to the first how. How do you get it? Well, non-typhoid salmonella infection has been linked to colorectal cancer. Additionally, infection-associated colorectal cancer might also develop via cell transformation to premalignant lesions. And the final how is it common? The incidence of colorectal cancer has been increasing in young adults over the past few decades. Furthermore, approximately 50 to 60% of incident cases of colorectal cancer in the U.S. are estimated to be attributed to modifiable risk factors. Let's touch on some relevant microbial populations. The gut microbiome plays a crucial role in the development of colorectal cancer. Specifically, bacteria, as mentioned before, like Akkermansia muciniphila and SCFA-producing bacteria, are associated with CRC prevention, while imbalances in microbial populations can contribute to increased risk. In addition, the human intestinal microbiome is primarily composed of firmicutes, bacteroidetes, actinobacteria, and proteobacteria. These microbial populations play essential roles in metabolizing indigestible food ingredients, synthesizing nutrients, modulating immune response, and maintaining mucosal integrity. Adding on to the previous slide, here's an illustration of a healthy versus an unhealthy colon. In disease states like colorectal cancer, there can be alterations in the abundance of healthy intestinal microbes leading to dysbiosis. Let's discuss the proposed mechanisms of either benefit or harm from microbiota. We'll start with the harms first. Harmful alterations in the gut microbiota, known as dysbiosis, can promote chronic inflammation and pathogenic changes in the microbiome profile and functions. Additionally, bacterial metabolites associated with CRC, such as secondary bile acids and SCFAs, can have detrimental effects on colon inflammation and can contribute to colorectal carcinogenesis. Now let's talk about the benefits to lighten the mood. The gut microbiota can benefit colorectal cancer treatment by modulating the immune and metabolic pathways affecting treatment outcomes and the survival times of patients. Furthermore, specific bacteria like Akkermansia muciniphila and Faecalibacterium have been associated with greater efficacy of PD-CD1 blocking agents, possibly by increasing recruitment of T cells to tumors and enhancing their functions. So how do we know all of this, and how is it tested? Well, a study conducted from Science Direct tested the effects of exercise on fecal microbiome composition and metabolites in sedentary adults. The study involved 2-week base analysis, 6 weeks of exercise intervention, and 6 weeks of washout. So the proof of the claim being made here is that the increase in fecal concentrations of short-chain fatty acids in lean participants after exercise, along with changes in butyrate-producing bacterial taxa associated with shifts in body composition. Here is additional information from the study. Integrated prospective studies will open new avenues for studies of intestinal microbiota and its manipulation in CRC screening, prevention, and treatment. Additionally, understanding the influence of the microbiome, diet, and environmental factors on CRC could lead to the development of new strategies for CRC screening, chemoprevention, and treatment. And finally, The findings from future research could potentially help in utilizing features of the intestinal microbiome for CRC screening and modifying them for preventive and therapeutic interventions. In conclusion, the key takeaway from this study is the importance of understanding the influence of the gut microbiome, diet, and environmental factors on the risk of colorectal cancer. It also emphasizes the need for prospective studies to investigate how these factors impact the gut microbiome, its metabolism, and its effects on CRC prevention. If you would like to read more about this study, visit ScienceDirect.com, where you will find further information that was associated with this video. Well, that's all from me. Thank you guys so much for listening to me ramble on, and I hope you enjoyed and are now further educated on colorectal cancer and its relation to the gut microbiome. I hope you all have a wonderful day.